Good afternoon, ladies and gentlemen. We're going to talk about this afternoon about the gene therapy was made by also our instructor from our Science and Technology and Society, Mr. Alfred A. Turga. This is our intended learning outcomes. First is to describe the gene therapy in its various forms. Second is to discuss the prevalence of gene therapy in daily life. And the third is to explore the opportunities that may be opened by gene therapy in the future. The question is, what is gene therapy? The gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. It is a technique for correcting defective genes responsible for disease development. Gene therapy is an experimental technique for the treatment or prevention of disease using genes. In the future, instead of using drugs or surgery, this technique could allow doctors to treat the disease by inserting a gene into the cells of a patient.
So now let's dive into the history and development of gene therapy. So in 1960, the concept of gene therapy was introduced. In 1970, Friedman and Robin, author of a paper in Science titled Gene Therapy for Human Genetic Disease, cite the first attempt to perform gene therapy. In 1990, William French Anderson is known as the father of gene therapy. The first gene therapy was given to a 4-year-old girl suffering from ADA deficiency.
The first approved gene therapy case at the National Institute Health, UK. It was performed on a 4-year-old girl named Ashanti De Silva. It was a treatment for a genetic effect that left her with an immune system deficiency. New gene therapy approach repairs errors in messenger RNA derived from defective genes. This technique has the potential to treat the blood disorder thalassemia, cystic fibrosis, and some cancers. Sickle cell disease is successfully treated in mice. 1992, Dr. Claudio Bordignon, working at the Vita Salute San Raffaele University Milan, Italy, performed the first procedure of the gene therapy using hematopoietic stem cells as a vector to deliver genes intended to correct hereditary diseases. 1999, death of G.C. Gelsinger in a gene therapy experiment resulted in a significant setback to gene therapy research in the United States. 2006, scientists in the National Institute of Health have successfully treated metastatic melanoma in the patients. This study constitutes one of the first demonstrations that gene therapy can be effective in treating cancer.
In continuation with the history and development of gene therapy, in the year 2007 to 2011, research is still ongoing and the number of diseases that has been treated successfully by gene therapy increases, such as the retinal disease, color blindness, and the adrenoleukodystrophy. And in the year 2011, medical community accepted that it can cure HIV, as in 2008, Gerald Hatter has cured a man from HIV using gene therapy.
We have two different types of gene therapy, depending on which type of cells are treated. First, we have somatic gene therapy, which is transfer of a section of DNA to any cell of the body that does not produce sperm or eggs. Effect of gene therapy will not be passed on to the patient's children. Somatic cell gene therapy. Therapeutic genes transferred into the somatic cells, for example, introduction of genes into bone marrow cells, blood cells, skin cells, and etc. It will also not be inherited later generations. At present, all research directed to correct genetic defects in somatic cells. Second is germline gene therapy. Transfer of a section of DNA to cells that produce eggs or sperm. Effects of gene therapy will be passed on to the patient's children and subsequent generations. Germline gene therapy is that therapeutic genes transferred into the cells or germ cells. For example, genes introduced into eggs and sperms. Next is it is heritable and passed on to the later generations. Next, for safety, ethical, and technical reasons, it is not being attempted at present.
Now, I will explain the difference between somatic gene therapy and germline gene therapy. Somatic gene therapy involves modifying or correcting genes in non-reproductive cells to treat specific medical condition. It doesn't affect an individual's germline, so the genetic changes aren't passed on to the future generations. Meanwhile, germline gene therapy targets reproductive cells aiming to introduce genetic changes that can be passed on to the offspring. Unlike somatic gene therapy, this approach could impact future generations by altering the individual's germ cells. It raises ethical concerns and is currently a subject to careful considerations and debate within the scientific community. Now there are two types of somatic gene therapy. First is ex vivo. Here the cells are then taken out from the body and grow in the laboratory. These cells are then exposed to the virus containing the desired gene, and then after recombination, the recombinant cells are returned to the patient. The second one is in vivo. Here the genes are transferred to the cells present inside the patient's body.
Basic process of gene therapy. Several approaches to gene therapy are being tested, including replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functional improperly, introducing a new gene into the body to help fight the disease.
What is a vector? Gene therapy works by introducing genetic material, or such as a transgene or nucleus, into the nucleus of the cell. The system used to deliver genetic material is known as a vector. Think of a vector as a microscopic delivery truck that transports packages or genetic material to specific locations or target cells. In general, a gene cannot be directly inserted into a person's cell. It must be delivered to the cell using a carrier or vector. Vector systems can be divided in two. These are viral vector and non-viral vector.
Viral vectors. Viral vectors are tools commonly used by molecular biologists to deliver genetic material into cells. This process can be performed inside a living organism or in a cell culture. Viruses have evolved specialized molecular mechanisms to efficiently transport their genomes inside the cells they infect. Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. Scientists have tried to harness this ability by manipulating the viral genome to remove disease-causing genes and insert therapeutic ones. Viral vectors in gene therapy. Lentiviral vectors, or LVVS, are a species of retrovirus. The most well-studied lentivirus is HIV, and scientists have used its blueprint to design lentiviral vectors for gene therapy. Lentiviral vectors have the ability to enter the cell and insert its genetic material into dividing cells, such as stem cells, and non-dividing cells, such as cardiac cells. Lentiviruses also integrate genetic material into the host genome. This integration means that the genetic material has increased durability, which allows for continued gene expression.
Adenoviral vectors. The first viral vector used in gene therapy was based on adenovirus, which is a virus that causes the common cold. Some ADVs were found to trigger strong, potentially dangerous immune reactions in patients. However, considerable research has helped to understand these immune responses, and ADVs have shown promise in the treatment of cancer. Further research in the use of ADVs is still being explored. The next is adeno-associated viral vectors. Research and creation of adenoviruses led to the discovery of AAVs in 1960s, and they are considered the standard viral vector used in gene therapy today. These type of vectors are the most studied and most frequently used for gene therapy and are currently involved in a wide variety of clinical trials. There are many different types of AAVs. Each type has a different property allowing them to target different cells, ranging from kidney cells to Each type has a different property allowing them to target different cells, ranging from kidney cells to neurons in the brain.
So, next up, the non-viral vector. So, non-viral method presents certain advantages over viral methods with simple large-scale production and low-cost immunogenicity, being just two. Previously, low levels of transcription and expression of the gene had non-viral methods at a disadvantage. However, recent advances in vector technology have yielded molecules and techniques with transcription efficiencies similar to those of viruses. So, there are two main ways of non-viral vectors. So, one of them is physical vector. So, physical vector allows researchers to directly deliver genetic material to target cells, for example, like using an injection needle. So, another example of a physical vector is the electroporation. So, it is a non-viral delivery method being studied to deliver gene therapy in clinical trials. With electroporation, scientists use pulses of electricity to cause temporary pores to form in a cell membrane. These pores enable gene therapy to be delivered into the cell where it can take effect.
Two main ways of non-viral vectors. Chemical vector. Use natural or synthetic materials that are compatible with the human body so they are not as likely to generate immune responses. These include fat molecules, polymers, and nanoparticles. Example, lipid nanoparticles. The leading non-viral delivery method uses lipid nanoparticles. LNPs encapsulate genetic material so that it can be delivered to target cells. Without a delivery system like an LNP, genetic material degrades quickly and cannot reach target cells. Because LNPs are quickly to manufacture, efficient, and scalable to the size of the material being delivered, they have many potential uses in gene therapy.
Pros. Provide option genes. Therapy can potentially cure someone of a disease, especially in the instances where no other than medications have worked. Next is only has to be given one time existing. Existing genes therapies are given as a one-time dose. This is appealing compared to some other treatment that require multiple doses. Next is long-lasting effects. Gene therapy has the potential to get rid of a person's symptoms for life. Gene therapy can be given many people a better quality of life. Next is a positive effects passed down through generations. If you remove a faulty genes from a parent, they won't transfer this gene for their kids. Rapidly changing technology. Gene therapy is able to exist because of massive advances in the technology over the past few decades. Since technology continues to advance, we can expect gene therapy to continue to improve. We can also expect more gene therapies to gain FDA approval.
If there's a pros in gene therapy, there can also be a cons, which are first, it is expensive. Gene therapy can be extremely pricey, making it inaccessible for some people. For example, Zolgensma, a gene therapy to treat spinal muscular atrophy. It is the most expensive medication in the US. It costs $21 million for a course of treatment. So, this
This is only accessible for those who has privilege or for those people that is rich and can afford this kind of treatment. Second, it is experimental. Gene therapy is relatively new and there's still a lot about it that we don't know. Right now, there are no guarantees. Exactly how successful gene therapy will be is still uncertain. So, as you can see, gene therapy is still new that there's a lot of things to know more about this therapy because it is not given nor guaranteed to be 100% successful. For this still needs a deeper knowledge and research for this to be able to work.
Potentially dangerous. There are some concerns that if gene therapy reaches other cells in the body besides the faulty cells, it have harmful effects. There's also concerns that reproductive cells could be changed without detection, and this could be passed on to people's children. Ethical issues. Some people have ethical issues with gene therapy. For example, some wonder who will decide what good and bad uses of gene therapies are. And while gene therapy makes us less accepting of people who are different, may cause infection. Some gene therapies use inactivated virus to carry healthy genetic materials into a faulty cells in the body. It's possible that this virus could infect the person receiving therapy.
In conclusion, theoretically, gene therapy is a permanent solution for genetic diseases, but it has several complexities. At its current stage, it is not accessible to most people due to its huge cost. A breakthrough may come anytime and a day may come when almost disease will have a gene therapy. Gene therapy have the potential to revolutionize the practice of medicines.